So, DNA is made up of two anti-parallel strands. One runs from 5' to 3' and is called the sense strand. The other one runs from 5' to 3' but in the opposite direction and is called the antisense strand. Both strands of DNA have a sugar phosphate backbone made up of a phosphate and a deoxyribose, which is a 5-carbon sugar. These then are each bonded to a base, otherwise known as a nucleotide. You've come across these before at GCSE. They're known as adenine, thymine, cytosine and guanine, or ATCG for short. Adenine will bind to thymine with 2 hydrogen bonds, and cytosine will bind to guanine with 3 hydrogen bonds. The binding of these together then creates like a spiral staircase, which then allows all your DNA to be compacted into the cells of your body. So, got to remember they're complementary base pairing. It's really, really important that we remember it's complementary. Your DNA has equal amounts of A and T and C and G, and this DNA makes up a code, which is all your genetic information, i.e. it's a blueprint of how to make you. So DNA, it's double-stranded, it's stable, it's found in the nucleus, primarily in the nucleolus of the nucleus, but it's too large to get out of the nucleus, and that's a problem because we want to do something with this genetic information. We want to make it into proteins, but to do that we've got to get it out of the nucleus and into the cytoplasm. So what happens here then is our DNA becomes transcribed into messenger RNA, otherwise known as mRNA. mRNA is shorter than DNA, it's single-stranded, it's quite unstable. Initially, when messenger RNA is made, it's made as pre-messenger RNA, and this has extra junk in it, so it gets spliced. So all the unnecessary information gets cut out of it, and it gets caps put on the end of it to make it a little bit more stable when it goes into the cytoplasm. But we don't need messenger RNA to last a long time in the cytoplasm. Once it's been translated, it can then be got rid of. Messenger RNA then can also leave the nucleus via these nuclear pores. It's only one strand, and unlike DNA, it doesn't have a thymine, it has a uracil instead. Again, this is something you have learnt at GCSE. So in the cytoplasm, the messenger RNA gets translated into amino acids. These get joined together by peptide bonds to make proteins. So this would be a really good time to go back and revise proteins. I want to point out as well, whilst I change my sheets of paper, that only one of you was able to answer the question of showing a peptide bond in your PPE. So it would be a really, really good time to have a look at it. So this is transcription. That's the first bit. So this is, we're going to go through transcription and translation. So as you can see, excuse my non-manicured hands, here's my double-stranded DNA with my complementary base pairing. Here is my nuclear envelope with my nuclear pore, and this is too big to get through here. So we need to make it smaller. So what happens then is that the double strand of DNA unwinds and unzips. If we think about this like a zip on a coat, and unzipping it then, we break the hydrogen bonds. So what happens then, if we go back and think about it, we've got our sense and our antisense strands. Our sense is what we want it to look like, and the antisense is the opposite one. So our antisense one acts as a template for the new DNA strand to be made, because we want our new DNA strand to look exactly like the new RNA strand, to look exactly like the DNA strand, So unzipping it, what happens then is we make this bubble, occur, and complementary free RNA bases now come in and base pair with the antisense DNA strands. The difference between RNA and DNA is the sugar. It's a 5-carbon sugar, but this time it's got a ribose in it instead of a deoxyribose. So if we have a look now, then we notice that if our DNA strand was to read adenine, thymine, cytosine, guanine, so A, T, C, G, our adenine now would bind to a uracil, not a thymine. This is the difference between RNA and DNA. Our thymine get an adenine, our cytosine would get a guanine, this time with 3 hydrogen bonds, and our guanine would get a cytosine with 3 hydrogen bonds. Okay? So, then there's a big enzyme. Okay? We don't need to know the structure of it for BTEC, we just need to know it exists. This enzyme is called RNA polymerase, and what it does then is it forms the phosphodiester bond between each one of these RNA molecules so that we end up with a strand. So this pencil line here that I've drawn is where the RNA polymerase has then bonded all these single RNA bases together to make a strand, okay? This strand then can detach from here, so we break these bonds down here again using RNA polymerase, and then we can leave through the nuclear pore into the cytoplasm. Now, I've written pre-messenger RNA here. So what happens then, this messenger RNA gets caps put on either end of it to make it more stable, and any junk that happens to be here, so any DNA sequence that doesn't necessarily make up the gene, it's there more as packaging, if you think about it as bubble wrap around the DNA, is then spliced out. You just need to know that splicing exists, you don't need to know how it happens. This messenger RNA then travels out of the nucleus, through the nuclear pore, and binds itself to small ribosomes which are found in the cytoplasm of the cell. Okay? So, the ribosomes then are found all over the cytoplasm, and they are the site of translation and the site of protein synthesis. So, ribosomes, they're made up of 2 subunits, which are then individually made up of ribosomal RNA, known as rRNA. The rRNA is really important to maintain structural stability for protein synthesis, and it also catalyses protein synthesis, so it catalyses the whole reaction. Okay, so that's the importance of ribosomes. So if you are asked in an exam question, where are the sites of protein synthesis, the answer is ribosomes. So translation then, what happens? So my messenger RNA will bind to the small subunit of the ribosome. Remember, there are 2 subunits, and it will bind at a codon. Now a codon is 3 base pairs, so 3 bases or nucleotides in a row. And it has a specific one, which is a start codon, which is AUG, adenine, uracil, guanine. Okay? So a transfer RNA now, so this is the final of our RNAs that we're going to learn about today, will then bind. And if we have a look, they have a specific shape. So this bottom bit down here has the complementary anticodon. So if the codon reads in one direction, the anticodon is the opposite to it. Okay, so it has an anticodon here. Then it has this funny T shape, which is where it gets its name from. And at the top of it then, it has an amino acid. That amino acid there is specific to the anticodon at the bottom. Okay, so in the cytoplasm of your cells, you've got lots and lots of transfer RNAs, all with specific anticodons with specific amino acids on them. You have 20 amino acids. So all our DNA then will encode up to 20 amino acids, and the 20 amino acids then bind together to make all the proteins. Okay, so this is a really good time to go back and revise your protein synthesis. So here then, I've shown in the green surrounding is the ribosome. So any one time there can be 2 codons within the ribosome, so you could bring in 2 transfer RNAs. Okay? So what happens is that the ribosome moves along the messenger RNA, and as it moves along it brings in these transfer RNAs at the top here with their complementary amino acids at the top. Okay? The ribosome then will also bring into play an extra enzyme, okay, and it's going to catalyze the formation of the peptide bonds. So we need to join these amino acids together. This is all just the structure to be able to bring these amino acids together. Okay, we're going to get a peptide bond by the enzyme peptidyl transferase. Okay, as the ribosome moves along and the amino acids get bonded together, the transfer RNA then drops off, goes back into the cytoplasm to pick up its complementary amino acids, so it can start the cycle all over again. Okay, so what we then end up with is a chain of amino acids all bonded together, and this is our primary sequence. So go back to what we were learning about October, November time, and you'll come back to your proteins. Okay, so when these get folded together, we make our secondary structure, and our secondary structure is our alpha helix or our beta pleated sheet. Then we get our tertiary structure, which is globular or fibrous proteins, and then for some proteins you would get the joining of protein subunits together, which is the quaternary structure. Hemoglobin is a really, really good example of all of this. Okay, so that is DNA, transcription and translation all done in one case. So what I want you to now do is have a look on the Show My Homework, and you'll see I've got just 2 questions for you to answer. If you can email those to me, your answer to me, that would be absolutely brilliant. I now want to introduce you to mutations. So we've talked about using our DNA sequence to make the proteins. The proteins are what our entire bodies are made up of, but sometimes the sequence of DNA gets a mutation, so this gets a change. Okay, this change could come about because of sunlight, it could come about food, it could have been a mutation due to an issue in mitosis or meiosis, it could have been an issue in DNA replication, so things we've looked at before. This could be a mutation that is just in your body, or it could be a mutation that has been passed on. So if it's one you've acquired in your body, in your lifetime, it's called an acquired mutation. If it's one that has been passed down from generation to generation, it's called a hereditary mutation. Okay? So there's lots of different types of mutation. So the simplest one is called a point mutation, and there are 3 types of point mutation. The first is called a silent mutation, where the DNA sequence gets changed, but the amino acid that it encodes for stays exactly the same. Okay. The second one is called a nonsense mutation, where the DNA sequence gets changed, and this time the amino acid gets changed, and what it does is it puts a stop codon in place, and this stops protein synthesis from occurring. Okay, so you end up with a truncated or shortened protein, and that could result in the protein then being denatured and absolutely non-functional. And the final one is called a missense mutation, where the DNA sequence gets changed, okay, and this causes a whole different amino acid. So here we've got a tyrosine, and it gets changed to something called a histidine. Now on the face of it, we've only changed one or 2 parts of this, okay, but actually what has happened then is we've got a whole new protein going into our protein sequence, so we could be changing the whole structure of that protein. So it may no longer fold how it's meant to fold. If this was an enzyme, this could affect its active site. Okay, so we don't know what effect this could have. So these are the simplest of the mutations, where we're just changing one or two of the bases, okay? Then there are other sections, so we could delete a whole section of the DNA. We could invert it, okay? So here you can see I've got a sequence A, B, C, D, E, F. Then I've got A, B, C, F, E, D, so I've just switched part of it around. We can get translocation, so this is like a whole chromosome now, not just a simple section of DNA, it's a whole chromosome. So here I've got an orange and a yellow chromosome, and what's happened now is we've got part of the yellow and part of the orange have just snapped off and changed with one another, okay? Now this, if it gets carried over to the next generation, then could have lots and lots of repercussions, because the coding of the genes on this chromosome and this chromosome are going to be completely different. And then the final one is something called polyploidy. So this means when we have extras. So we could have just an extra copy of a chromosome. So Down syndrome is a great example of this, because they have 3 copies of chromosome 21. Or we could have a whole new set of chromosomes, okay? Now, humans really struggle with having extra chromosomes or extra sets of chromosomes, but there are lots of organisms out there that actually having extra sets doesn't really affect them. But at the end of the day, we're interested in the human part, so for us, this causes lots and lots of issues, okay? So, the reason I've given you this piece of information is I want you to have a look at the Show My Homework now, and I've set you a task to find out about a couple of disorders associated with mutations, okay? I want you to find out about what causes them, what are the symptoms of them, and what is the treatment, if there is any treatment, okay? Right, I hope this has been useful. Please email me any questions. My email address is all over all your Show My Homework stuff. I hope you are all staying really, really safe. Bye for now.